Hi, I'm Miranda. My project for Computing for Global Challenges was to conduct a literature review on quorum sensing, and particularly quorum sensing as modeled through computational means. So just some background. Quorum sensing is a mechanism by which bacteria decide whether or not to engage in collective behavior. And they do this by secreting small chemicals called autoinducers that accumulate in the extracellular environment. And just as sort of an example system, the bacteria Vibrio fischeri uses quorum sensing to regulate bioluminescence in its squid host, the Hawaiian bobtail squid. And as you can see sort of at the top there, when Vibrio fischeri is at a low cell density in the squid, it does not bioluminesce, because if there are only like one or two bacteria that are engaging in bioluminescence, it's not going to light up the whole squid. And therefore it's not worth it for the bacteria to invest in producing the enzymes required for bioluminescence. But once a certain cell density is achieved, as you can see in the diagram there, autoinducer accumulates outside the cell. It gets imported into the cell. It binds with a receptor that then upregulates transcription of the luminescence genes. And particularly important are LuxA and LuxB, which then combine to produce luciferase, which is the main enzyme involved in bioluminescence. And so, therefore, at a high cell density, all of the bacteria, at least in theory, are bioluminescing and the whole squid lights up. So this isn't the only behavior that can be regulated by quorum sensing. Biofilm formation, exoenzyme secretion, virulence factor production, all of those things can be regulated by quorum sensing. However, any sort of cooperative behavior like that comes with the problem of free riders. So one thing that we were looking at is, why is cooperation worth it when, say, a bacterium that doesn't bioluminesce at all can still live in the squid and derive the benefits without also paying the cost of producing luciferase? So some different mechanisms by which bacteria can prevent cheats from overriding the population are through kin or kind discrimination, where the bacteria recognize other bacteria that are cooperating and make sure they only direct the benefits to other cooperating bacteria. And that could manifest, for example, through a punishment system. For Vibrio fischeri, we think that the squid itself actually punishes bacteria that aren't cooperating, but other bacteria are sometimes able to punish each other, maybe through their quorum sensing molecule is a toxin, and part of the gene that's being upregulated is the antidote to that toxin. And if the cheating bacteria don't respond, then they're killed off. Or maybe cheaters are intrinsically better suited to survival and will increase in proportion in the population. But because bacterial populations are disturbed so much and so frequently new bacterial populations are forming, bacterial populations that are just cooperators may be able to are able to grow larger by virtue of the fact that they're cooperating and offset other populations in which cheats are overriding those cooperators. And that's called Simpson's paradox. But anyway, the end goal of this project was to develop a common minimal model that would allow us to see how quorum sensing emerges as an adaptation in bacteria where previously cooperation was not necessarily occurring. So in our design, and not all of it is in this model up here, but we have 6 states, and 4 of those states are sort of basic actions that any bacteria would perform, like moving or dividing or dying or just absorbing nutrients. But we also worked in a cheap extracellular compound, which does have a small benefit to the cell, but can be produced without much cost to the cell as sort of a potential molecule that would become an autoinducer theoretically in a real-life population, and then also an expensive extracellular compound that would act as our public good, where it's very difficult for the bacteria metabolically to produce it, but as long as it stays around the cell in a high enough concentration, or other bacteria are producing it in a high enough concentration, it can still provide benefits to the cell, and higher benefits than just the cheap extracellular compound would as the autoinducer. That's all. Oh, wait, sorry. And these are references for this PowerPoint. Those weren't all of the references, or all. There certainly is not a comprehensive list of the literature that I reviewed, but that was the information relevant to this presentation. But